Next on our virtual travels is the Finger Lakes region of upstate New York. The Finger Lakes region has long been a destination for its hiking, waterfalls, fishing, and of course, wine production. The Finger Lakes region, which comprises 11 major lakes and many more smaller ones, is home to over 130 wineries. It's a much cooler and more humid climate than wine regions out west, which makes it ideal for making Rieslings, Cabernet Francs, and other European-style wines.
It's a fascinating region because historically people didn't think that the European wine grape would do very well here. But then probably about 60 years ago, more or less, some adventurous winemakers started growing vinifera grapes and realizing that they could make really good wines here. And especially our region is quite similar to Germany. And so the wine varieties that you think of as German and Austrian, they tend to do quite well in the Finger Lakes region of New York.
That's Lance Cadle-Davidson, a research plant pathologist at ARS's Grape Genetics Research Unit here in Geneva, New York. Cadle-Davidson mentioned that, in addition to the traditional European grape varieties grown here, there's also some unique grapes that were developed in this region.
Cornell University has for more than a century had a wine grape breeding program here in Geneva, New York, and they've released a number of varieties that have taken off across the country. Traminette being one of the famous ones, which has the kind of traditional Gewürztraminer flavors and aromas. It's been amazing to watch the wine industry grow over those years.
Growing wine grapes in a humid climate like the Finger Lakes can have its challenges, mainly dealing with different fungal diseases. Because many of the grapes grown here originated in Europe, the grapes can be highly susceptible to certain pathogens that are not common in their native regions. Consequently, grape growers may have to spray their vineyards with fungicides between 10 to 15 times a year.
We've been working hard with grape breeders across the U.S. to develop disease-resistant varieties that don't require as many pesticides. And the varieties that we're developing could have a 90% reduction in pesticide use compared to the European varieties and still have an excellent fruit quality and wine quality. But one of the challenges for a grape breeder or really any scientist is when we measure traits in the natural environment, out in a vineyard, there's a lot of uncontrolled variation in an experimental design. From across a field, you might have variability in disease pressure, you have variability in the genetics of the pathogen and which isolates land on which plants.
With such variability in the fields and the seasonal changes occurring year to year, it was hard for researchers to study and analyze the effects these diseases were having on the vines. That also made it difficult to recommend to the grape breeders which genes or traits to focus on to make them more disease resistant.
So what we did about 15 years ago is bring the research into the laboratory. We'd sample leaves from a vineyard, from a breeding program, from across the country, it didn't matter. And we'd process those leaves as 1 centimeter leaf discs, the size of a nickel or a dime. And in controlled environments, we inoculate them with the pathogen, and we can control how many spores we put on the plants. We can have them all in one baking sheet kind of environment. And then we could use microscopy to have very detailed quantification of exactly how much disease grew on each grapevine. In our case, we're thinking about disease resistance genes. And so we could have not only like the light switches that have a strong effect of resistance either being on or off, but we could do dimmer switches. And we have like just turn the dial up 10% and have a little more disease resistance, or add that with one gene that has 30% increase in resistance. This was a game changer, but the problem is we ran into a time crunch.
This new process involved hours and hours of manually collecting data from the leaf experiments, sometimes making over 1 million observations from a microscope. Each experiment could take a month or two to analyze and collect the data, sucking loads of time from the researchers.
Beautiful quality of data, great discoveries, but it was too slow. What we realized was we could automate the process. What we did is we combined new sensors that were high resolution cameras with illumination so we could see the fungus easily from LED lighting, with artificial intelligence to quantify disease, and robotics to capture images quickly in an automated system.
Okay, for those who like to geek out about science, I'll let Cadle-Davidson explain how the robot works. You can also see the robot in action by clicking on the link in the description.
Imagine a baking sheet with 351 1 centimeter leaf discs arrayed across that sheet in a grid, which is about half an acre of grapevines because grapes take huge space, right? So we can put it onto this Cartesian robot. It's an XYZ robot. It moves the sample in an x-axis from one sample to the next, and then the y-axis to the next row, etc., until it's imaged every single leaf disc on the tray. And it uses the z-axis to focus, right? Because at a microscopic scale, a leaf is actually not a flat surface. It has a lot of topology and undulations. And so we have to capture multiple pictures in the z-axis and flatten them into one image in order to get a perfectly focused image. And once we have the images, it's still too many for a human to look at and objectively quantify disease severity. So we use artificial intelligence, and specifically convolutional neural networks that were developed in the past decade to quantify how much disease is on every single leaf disc. And we actually find that using artificial intelligence to quantify the disease in the image is more accurate than a human expert sitting at a microscope day in, day out.
The robot is called Blackbird, and it works similar to a 3D printer, scanning and detailing all angles of the subject and then storing that data for printing, or in this case, processing. Cadle-Davidson currently has 8 of these robots in his laboratory, and the results are truly amazing. In just 6 hours, the robots can scan and quantify the same amount of data that it took researchers an entire year to complete.
This has completely revolutionized the kinds of research questions we can ask and the throughput with which we can make discoveries. An example of this is, in the past 2 years, we've discovered about 25 new sources of disease resistance in grapevine with genetic markers that can predict which vines are going to be resistant and which vines are going to be susceptible. In the previous 30 years of research internationally across all diseases of grapevine, only 60 such discoveries have been published. So it's really revolutionized our science.
The Blackbird robots have not only sped up the evaluation process, but in turn has helped wine grape breeders all over the world determine which traits to identify for breeding more disease-resistant grapes.
They send us leaf material, and we crank it through the Blackbird system and give them the data along with the genetic data and predictions of which plants they should keep and which they should discard, which resistances look really good, and which ones are not as interesting and maybe could be deprioritized. It's been a really great partnership because that allows us to look at many, many different sources of resistance that we otherwise couldn't look at by ourselves.
As you might expect, Blackbird can evaluate just about any type of material, which is why they're building more robots for other ARS labs across the country. And Cadle-Davidson is receiving much more than wine grape leaf samples to image.
We've imaged a lot of interesting things I never thought that I'd be working with, like fish embryos and fish eggs, or nematode microscopic worms. We've looked at tropical nut borer beetles from Hawaii and all kinds of different stuff.
Getting back to the wine grape research, more than a decade ago, Cadle-Davidson decided to bring the research from the field into the lab. Now, he plans on taking his Blackbird back out to the field to identify the presence of pathogens in real time.
We now have vineyard robots that drive up and down the rows right next to the vines, and they're imaging the vines to quantify disease there. This has been very successful for breeding programs where some of the vines might be completely hammered by disease, have 100% disease severity, and others 0%. It's not yet game ready for commercial vineyards because there you really need to find the needle in the haystack, to find really rare disease and be able to spray it or spot treat it before it becomes a bigger problem. But that's one of the things that we're working on in collaboration with Cornell University and other scientists across the country. In creating the Blackbird initiative, the goal is to create the world's largest ecosystem for biological microscopy data. And we think that with the current scale of what we're envisioning, we'll be able to do that in the next 3 years.
We're going to make a jump here, from the future of robots to the past with regenerative farming. In our next episode, we'll see how enhancing these classic farming practices can help us build a sustainable future for wine growing.